Hello everyone and welcome back to Willow's Notes. In today's video, we will learn how to identify whether two genes are on different chromosomes or are linked, meaning they are on the same chromosome. Let's start by looking at the givens. Here we have a wild type gray body, normal wings fly, crossed with a mutant pink body, short wings fly. As you can see, 100% of the first generation F1 have gray body and normal wings, which means gray body and normal wings are dominant traits, which immediately eliminates option D that claims that the alleles for pink body and short wings are dominant. Next, a hybrid F1 fly is crossed with a mutant fly, what we call a test cross. Before we look at the given results, let's first talk about the two possible scenarios. The first scenario being that the gene for the body color and the gene for wing shape are on two different chromosomes. So using the FOIL method, let's determine the gametes for the gray fly. F first of each pair, so we're going to place capital G, capital N. O will be capital G, small n, I, small g, capital N, and then L for the last of each pair, which is small g, small n. As for the pink fly, I think that's quite easy because all the gametes, 100% of the gametes are going to be small g, small n. Next, we fill in the table and we get the genotypes of the offspring. As you can see, we got a ratio of 1 to 1 to 1 to 1. The second scenario is that the two genes are on the same chromosome. If alleles G and N are on the same chromosome, then in the gametes we will have two possibilities. We will have gametes carrying the capital G, capital N chromosome and gametes carrying the small g, small n chromosome. These two will not even exist. Then we'll have two phenotypes which are the same as the parental genotypes with a ratio of 1, 1, 0, 0. Now let's go back to the results of the question we have and look at the numbers or ratios of the offspring. These expected numbers are what we expect to see, considering that the genes are on different chromosomes. Okay, like we just saw in the first scenario, we get a ratio of 1, 1, 1, 1. If we calculate the ratios for observed, we can clearly see it will not be 1, 1, 1, 1. How do we calculate ratio? Very simple. Just divide all the numbers by the largest number. For example, 720 is clearly the largest number, right? So 720 divided by 720, that's 1. 700 divided by 720, you'll get 0.97, which is approximately 1. So that's another ratio of 1. 150 divided by 720 is 0.2. And finally, 130 divided by 720 also gives us 0.18, which is roughly 0. So the results show a ratio of 1 to 1 to 0, 0, instead of the 1 to 1 to 1 to 1, which implies that the two genes in this question are linked, meaning they are on the same chromosome. To this point, we have everything we need to answer the question, which is clearly option B. The genes for the two traits are on the same chromosome. This option also says that crossing over occurred between them. So let's talk more about that. When we wrote the ratios for observed, we agreed it's 1, 1, 0, 0. However, in reality, these two genotypes did exist. 150 flies were gray with short wings. 130 flies were pink with normal wings, so they weren't actually 0. These two phenotypes did exist. The explanation is that crossing over has taken place. Crossing over takes place in prophase 1 of meiosis 1, where homologous chromosomes exchange sections of DNA. If you wish to learn more about crossing over and meiosis, I will link my video in the description box. These phenotypes are called parental phenotypes because they are identical to the parents. However, these phenotypes are referred to as recombinants because they have a trait from one parent and another from the other parent. For example, these 150 flies, they have gray body but short wings. Okay? So the gray body from one parent and the short wings from the other parent. These 130 flies over here, they have pink body but normal wings. These recombinants are the result of crossing over. If you'd like to know how we calculate recombinant frequency and draw linkage maps, then stay tuned for the next video. Don't forget to like and subscribe, and I'll see you in the next one. Bye!